Okay, so now we look at the chlorophyll molecule. So it's a pigment. Chlorophyll actually is a pigment. So this pigment actually absorbs light most strongly in the blue-violet portions of the electromagnetic spectrum, which is somewhere about like 400 to 500 nanometer regions. You know, it's about 400 to 500 nanometer regions where you have the violets and the blue spectrum there. Then also followed by the red portions. Red portion is something about like 600 to 700 nanometer. Okay, right now it's 600 to 700 nanometer, where it's the orange red regions there. Okay. Conversely, it's a poor absorber or the green and near green portion of the spectrum, right? Somewhere near to the 500 to 600 regions. Yeah, yesterday we have seen the absorption spectrum, right? Or no, not that yesterday, but on Tuesday. Monday, right? We have seen this absorption spectrum where very minimum absorptions at the green region. So that's why the plants look green, because they do not absorb this pigment. Chlorophyll. Chlorophylls, they do not absorb the green color. They reflect the green color. That's why what we see is a green. Are you clear? Okay, so they give the green color property of the chlorophyll containing tissues. Okay. So the chlorophyll molecule, if you look at the molecule, this molecule actually contains the hydrophobic phyto chain and also the porphyrin ring with the magnesium at the center. So magnesium is very important for the chlorophyll molecule because the photoexcitation actually takes place at the magnesium there. Okay, so magnesium will constantly give out the electron. Are you clear? When the magnesium gives out the electron, magnesium actually charged positive. Okay. So two types of chlorophyll exist in the photosystem, the green plant, which are chlorophyll A and chlorophyll B. And one thing that we need to know, chlorophyll A, they have the possibility to become the reaction center. Okay, there's possibility. But with term and conditions, they must have the peak absorbance at 680 nanometer for photosystem 2 and also 700 nanometer. Very big absorbent at 700 nanometer for the photosystem 1. Okay, so you compare the structure here. That's how we say that you have the hydrocarbon tail together with the porphyrin head. So this porphyrin head looks similar, right? This structure in the hemoglobin. The heme group of the hemoglobin also have this porphyrin head. Are you clear? Okay. So now center, instead of Fe 2+, in the heme group, we have the magnesium at the center here, magnesium. So hydrocarbon tail here, can you see that a long hydrocarbon tail? So this hydrocarbon tail is very important to interact with the phospholipids bilayer because the thylakoid membrane is a membrane. Okay, thylakoid membrane, again, remember thylakoid membrane is a membrane. So means that also consists of phospholipid bilayer. So how this chlorophyll molecule can anchor themselves in this phospholipids bilayer, basically they depend on this hydrocarbon tail that form the hydrophobic interactions with the phospholipid tail. Are you clear? So therefore anchored, embedded at the cell as this thylakoid membrane. Okay, clear? Clear. Now what make chlorophyll A and B slightly different is the functional group attached to this side. Can highlight, okay? Your CH3 is a chlorophyll A, CHO is a chlorophyll B. So that's why chlorophyll A and chlorophyll B, they are quite near. Are you clear? In terms of their absorption spectrum, but they're slightly different, okay? So chlorophyll B molecule is similar to the chlorophyll A. They have slightly different structure, so that's why they have slightly different wavelength for them to absorb in the red and blue part of the spectrum. And chlorophyll B only can act as the accessory pigment. No way for them to become the primary pigment or reaction center. Okay, clear? Accessory pigment. So the last part, we look at the keratinoids. So there are over 600 known keratinoids. Can you see that, guys? They have 600 known keratinoids. It's not one type. Keratinoid is a big group of pigment. Like chlorophyll is a big group of pigment as well. Chlorophyll A, chlorophyll B. Are you clear? Here we have 600 known keratinoids. We can actually divide them into two classes. One is called Centrophile, which contain oxygen elements. or keratins, which purely hydrocarbon and contain no oxygen. Keratins, they give the orange color. Keratins, they give the orange color of the carrot. Okay? And it's very, very important. This keratin is the precursor for our body to make vitamin A. Okay? So those orange color like tomato, okay, like carrots, they are rich in keratinoids, the pigment. Okay? So this pigment only absorb where? Only absorb at the blue-violet regions. Are you clear? So therefore, the green together with the green color together with the red reflected. So both reflected, so we will perceive this light intensity to our eyes as orange or yellow color. Are you clear? Okay, so write down for these keratinoids, they only absorb violet to a little bit of green lights, somewhere 400 to 550. The remaining one, they don't absorb, they reflect. So that's why it gives us a yellowish, orange, or even the red color. Okay, because they absorb mainly from 400 to 550, where again is a violet-blue region. Okay, so it acts as accessory pigments. No way for them. Okay, no way for them to become the primary pigment because they are not chlorophyll A. This one you have to know. And what is their role? Their role, they absorb the light energy and pass this energy to the primary pigment chlorophyll A in the reaction center. Now, I put this a bit confused. It's not all chlorophyll A. Yeah? Okay, I didn't put it in detail. This chlorophyll A. Okay, this chlorophyll A molecules, okay, must be the one have the peak absorbance at 680 or 700. Then they only can become the reaction center. So with this, I have done, okay, for this pigment, a photosynthetic pigment.